This is a research study titled CB1-Dependent Long-Term Depression in Ventral Tegmental Area GABA Neurons, a Novel Target for Marijuana by Lindsay Friend, Jared Weed, Philip Sandel, Teresa Nerfer, Isaac Oslin, and Jeffrey G. Edwards, critiqued and analyzed by Colleen Valentine. Drug addiction is a rising issue in the U.S. About $193 million is spent annually on costs related to illicit drug abuse. The theme that is present through almost all drug abuse is the effect that they have on the alteration of the VTA circuitry and function. Drugs of abuse enhance DA levels more than the normal release of rewarding behaviors. Dopaminergic cells are utilized in motivation and learned reward behavior. Cocaine induces LTP of excitatory input to VTA-DA cells, and these changes are possible causes of addiction behavior and the compulsiveness to self-administer drugs even after mitigation of acute DA enhancements. ECBs mediate many forms of plasticity, in particular LTD. These ECBs have been characterized as lipid-based signaling molecules that induce plasticity in the brain, inducing the VTA. ECBs have two active receptors known as 2AG and anandamide, which activate CB1 and TRVP1. THC has a negative effect in adolescence because it causes decreased cognition, abnormalities in the reward pathway, and increased probability of substance abuse. There has been an increase in pathological addiction known as cannabis use disorder, which is leading to memory deficits. Cannabis use disorder is in the DSM-5 and is the most prevalent drug use disorder in the U.S. There is still absent data of the drug's modification to the excitatory synapses onto the VTA GABA neurons. There must be action to study these effects because a decrease in GABA cell activity reduces inhibition to DA cells and thus enhances the release of DA. The goal of the study was to identify the mechanism of plasticity of excitatory inputs onto the VTA GABA neurons and determine the influence of THC on this plasticity. The animals that were utilized in the study were male juvenile adolescent age CD1 GAD67 GFP knock-in mice produced by the Tamakaki lab and were used so that GABA cells in the VTA could be positively identified using fluorescence. For slice preparation, the mice were anesthetized with isoflurane and decapitated with a rodent guillotine. Brains were immediately removed and sectioned horizontally on a vibratome at 300 micrometers using an ice-cold sucrose-based cutting solution. After sectioning, the slices were placed in an oxygenated ACSF. Recordings began at least 1 hour after cutting. The slices were placed in the recording chamber and bathed with oxygenated high-divalent ACSF along with 100 micrometers of picrotoxin to block GABA currents. GAD67 GFP positive cells were selected for recording from the same location in the VTA as in a previous study. Plasticity was induced using two stimulations at 100 Hz for 1 second, 20 seconds apart, or 5 Hz for 3 minutes. THC dissolved in ethanol was administered via intraperitoneal injection at 10 milligrams per kilogram with the final injection of volume of 50. Recordings began 24 hours following the final injection. GAD67 GFP mouse model was utilized to try and positively identify VTA GABA neurons by expressing GAD67 GABA cells with fluorescence green. A 5 Hz or 100 Hz high-frequency stimulus was applied to voltage-clamped GAD67 GFP positive GABA neurons in the mouse VTA in order to determine the ability of VTA GABA cells to exhibit synaptic plasticity of their excitatory inputs. The 5 Hz stimulation produced LTD of EPSC amplitude in about 30% of cells, but a more consistent LTD following the HFS was in about 85% of GABA cells. To determine whether the plasticity was postsynaptic or presynaptic, a coefficient of variance, PPRs, and failure rates were analyzed. Coefficient of variance and PPRs are an analytical means to examine data comparing evoked EPSCs during baseline with those after HFS. Coefficient of variance examines variation around the mean, which is decreased when LTD is presynaptic. PPRs examine the ratio of second pulse divided by the first. As presynaptic LTD depresses the first pulse more than the second pulse, an increased PPR is indicative of presynaptic plasticity. Increased failure rates also suggest a presynaptic site of action, as it is more difficult to induce transmitter release after HFS. Whereas individually these results are suggestive of presynaptic LTD, collectively these data strongly indicate a presynaptic mechanism for the observed plasticity. Additional controls demonstrate no effective vehicle exposure on baseline and that currents recorded from our glutamatergic is a control of THC bath application DMSO for 2AG, AM251, and MPEP. Among the candidate receptors known to bind ECBs and induce plasticity are TRPV1 and CB1. The TRPV1 antagonist capecitabine did not block HFS-induced LTD, suggesting that TRPV1 is not involved. However, application of the CB1 antagonist AM251 effectively blocked LTD. suggesting that CB1 is required for HFS-LTD. In Figure A, the TRPV1 antagonist capecitabine failed to block LTD, suggesting that TVRP1 is not involved in this HFS-LTD. In Figure B, the CB1 antagonist AM251 significantly blocked LTD. We applied DHPG in the presence of CB1 antagonist AM251. AM251 blocks most of the DHPG-induced depression, which is significantly different from DHPG alone as in Figure F. DHPG-induced depression was 16.5% with AM251 present and 47.1% in its absence at 10 to 15 minutes after DHPG application. Blocking HFS-LTD by postsynaptically inhibiting 2-AG synthesis, as in Figure D, suggests that the 2-AG involved in HFS-LTD is made directly by the GABA cell. To further confirm that ECBs are being produced postsynaptically in GABA cells, we examine an additional type of plasticity unique to CB1, DSE. We observed a significant depression following a 10-second depolarization to 0 millivolts, which was surprisingly persistent throughout the duration of the recording, which is present in Figure G. AM251 blocked this depression demonstrated in Figure G, suggesting that VTA GABA cells can produce the ECBs that can bind to CB1 in this type of plasticity. Research showed a novel CB1-dependent LTD of excitatory inputs to VTA GABA cells, which can be modified by intraperitonially injected THC. HFS-LTD is induced by CB1 activation that is mediated by postsynaptically produced 2-AG, formed by DAGL via mGLR5 activation. THC induces synaptic depression of excitatory synapses onto VTA GABA cells, which, applied acutely, occludes it when injected chronically. Therefore, this novel form of plasticity has the potential to influence acute reward and long-term potential addictiveness following THC exposure. It reported that using an HFS protocol depresses EPSCs of VTA GABA neurons in a CB1-dependent manner. This plasticity was presynaptic as 1-CV2 decreased, PPR increased, and failure rate increased, which collectively strongly suggested presynaptic expression. The data highlights that there are likely more ECB receptors at this synapse than just CB1, as R-methanamide and DHPG produce a larger effect than CB1 agonists alone, although CB1 is necessary for HFS-induced LTD. The former point was confirmed by the fact that AM251 only partially blocked DHPG-induced depression, also suggesting that mGLR activation is indeed tied to CB1-induced depression. Forms of mGLR-dependent LTD are also a feature of VTA-DA neurons. The mGLR1 induces LTD at excitatory synapses onto DA neurons, which reverses cocaine-induced strengthening at excitatory synapses mediated by exchanging GLR2 lacking AMPA receptors for those containing GLR2. GLR1 could still be involved in a postsynaptic or another plasticity form in VTA GABA cells due to the large DPHG-induced depression noted. mGLR5 is required for presynaptic CB1-independent LTD. DHPD inducible ECB or CB1 dependent LTD at inhibitory inputs to DA neurons was demonstrated where cocaine enables a normally inefficient stimulus to evoke LTD of iPSCs, whereas in VTA DA cells, CB1 induces iPSC LTD. In VTA GABA cells, CB1 induces EPSC LTD. Therefore, our data demonstrates that mGLR CB1 dependent plasticity is a feature shared by both VTA GABA and DA cells. The study covers an important topic in society that is increasing in importance every day because of the increased legalization of marijuana in the U.S. Marijuana is the most commonly used illicit drug, and its implications on the brain have not been researched thoroughly. The study had smart initiative to look at the effects of THC because that is the ingredient in marijuana that demonstrates addictive behaviors. It would be interesting in the future to see what other effects or changes in other areas of the brain that the use of THC elicits. The study did a good job at narrowing down what aspects of the VTA are affected by THC and trying to determine if the synapses are postsynaptic or presynaptic to the effect of THC, because that can help to determine if marijuana causes an increase or decrease in a hormone. It was also interesting to learn that THC has inverse effects on GABAergic and dopaminergic synapses to each other. I would have liked to have seen more research on how marijuana as a whole had an effect on the mice while they were alive, or to see it on a different animal model, rather than determining the effects from just the brain slices, because it is hard to determine depression from just a biological standpoint, along with the idea that changes in gender also have an effect. There is still much research being done on depression to determine if it is just a biological change or behavioral change. This journal was found on the Journal of Neuroscience website.